Hello everyone, welcome to this video presentation. Now this video is accumulation of all the things we learned all throughout the semester and their eventual conclusion. Now at the beginning of the semester we were given two sets of samples. One was the control, which was grown under normal circumstances, and one was the treated. Now both of these samples were E. coli samples. The goal was to find out what happened to those treated samples by analyzing their proteome. This was done by traditional different tools and techniques that are associated with proteomic based analysis. Now let's get to it. The first part of this experiment was to get the protein from the samples. We did the cell lysis, the digestion with tryptic enzymes. Then what we got at the end of that was a spectrum, or like several thousand spectrums from those proteins. Those spectrums in themselves don't mean much unless they are associated with certain peptides or proteins. That's where this software comes along, PixX. What Pix does is they assign a peptide to a spectrum, and those peptides can be mapped to a different protein. Now they use different statistical based analysis to keep scores to this protein. In most cases, the FDR is set to less than 1%. The FDR is false discovery rate. So when the software is assigning a score to a spectrum, they have a threshold which they call FDR value. Now, under these thresholds, we can be certain that these peptides are 100% matched to those spectrums. And so you can see that the curve is gently getting forward as the FDR value rises up. It means more protein, the more proteins you have, the more chances of them being a false positive. Now we took the samples which were less than 1% FDR values. So after all that, we have a list of proteins that were generated from the spectrums we were given from the mass spectrometer. But now those list of proteins in themselves don't mean anything unless we clean up the data a bit. The cleaning up process can be done by either Excel or SPSS or any other statistics-based tool. So what they will give us is a list of protein that's present only in the treatment and the list of protein that's present only in the control. Simultaneously, we can also perform an analysis where we can find which proteins are being expressed in a more abundant form in the treatment and less abundant in the control, which protein are being expressed more and which protein are being expressed less. Now, once we have that information, we need to find out what those proteins do inside the cells, which part of the metabolic process they're under. Now to find this, we can use either UniProt or Panther. What Panther does is they categorize the proteins in separate sections based on their functions, but at the end they always use the UniProt database as well. So the Panther is useful when you have like a large massive data set, since they can categorize and you can select based on those specific category. So since we are using a small set of data and we couldn't get that much protein out of it, I opted to use UniProt because that's pretty user friendly compared to Panther. Now, once you have that protein, once you have that function, you need to find out what they do in that metabolic process. So you need to find out the pathway. Now you can use string or cake database. What string does is string lets you find out which protein are interacted with what protein as well. So you can find out like a bunch of different proteins. They are simultaneously working together for a bunch of different functions. Now string is useful when you have a huge massive data sets. Since we don't have that, I opted to use cake pathway team. So now from the information I got from UniProt database, where I can see that this protein is generating is proteins involved in this pathway. Now I can analyze that pathway in cake pathway database, and then I opted to find out what that pathway means. Now this is important because when the cell in under certain stressful conditions, they will activate certain pathways, certain metabolic process when compared to the normal condition. By analyzing those pathways, we can infer what was done to the treated samples, which was our goal in the first place. The following two proteins I'll be talking about were only found in the treatment group and not in the control group, which means they were significant in our analysis, because whatever the treatment was done to the sample, it was causing them to express that protein involved in that particular pathway. Now let's get to it. Now, I found two proteins which were involved in the lysine biosynthesis pathway. The first one is the DAP-E protein and the second one was the RGP protein. Now, these two proteins, we can see that catalyze certain different reactions within that biosynthetic pathway. Now, the reason for talking about lysine biosynthesis out of all the pathways that we can go through is because lysine biosynthetic pathway is found to be involved when the pro cells are under tremendous oxidative stress. So this is one type of the defense mechanism when there's like an accumulation of reactive oxygen species. Now lysine can be involved in other pathways as well. This is an important amino acid, so it can be for energy generation or survival or any other defense mechanism. Now, the reason why I'm talking about oxidative stress in general is it will be clear in the later section of the video presentations. Another protein that was found only in the treatment group is the pyroxidal kinase. Now, this protein is involved in several different steps in the vitamin B6 metabolism pathway. Another name for vitamin B6 is pyridoxine. Sorry for that. Now, this vitamin B6 can be involved in the virulence factors of the bacteria, but get this is also a really good efficient oxygen quenchers for singlet oxygen. Singlet oxygens are the reacted oxygen species. So when it means these are the things that causes oxidative stress. So now for the bacteria to express the proteins that are specifically involved in the vitamin B6 pathway means they have a tremendous accumulation of reactive oxygen species within their cells. Now, reactive oxygen species means oxidative stress. It means it can infer that the bacteria are under oxidative stress. Now, let's just talk about the protein that was being upregulated in the treatment group and staying normal in the control group. One of the protein was involved in the citric acid TCA cycle. TCA cycle is an important mechanism of generating ATP from glucose. And the protein I found was PRDPE, which is involved in the process of making isocitrate from citrate or oxaloacetate. Now we can see that this protein was almost twofold increase in regulation compared to the control. It means that the TCA cycle was being upregulated in the treatment. Cycle can be used for generating energy, but get this is also involved in a defense mechanism against oxidative stress. Now everything comes back to this one particular problem, that is oxidative stress. Every evidence that we found that can be commonly linked to this particular problem, which means the bacteria, whatever the bacteria was treated with, it was causing them tremendous oxidative stress. Now these two evidence alone cannot be responsible for giving a proper conclusion. So let's talk about what the protein was being downregulated. One protein that I found was being downregulated was GLPC, which is involved in the glycerol metabolism, which is another pathway for degradation of glycerol. Now you can see some of the research that was done before, how the glycerol becomes there's like an abundant increase of glycerol inside the cells when the cells are under oxidative stress, which means the reactive oxygen species causes the pathway for glycerol metabolism to block them, and which in turn causes an increased amount of glycerol within the cells. So all of the pathways I've discussed throughout this video presentation point have only one thing in common. They are all active when the cells is facing oxidative stress. Now, oxidative stress means there's like a high number of oxygen-free radicals or singlet oxygen molecules inside the cell which damage the DNA, and that damage causes a cascade of events which lead to the pathways being active, inactive, upregulated, downregulated, some of the evidence that we just saw. One of the most common methods of giving oxidative stress to cells is UV radiation, then there's photooxidation, but most commonly used is hydrogen peroxide treatment. So throughout this analysis of proteins in cells, I concluded that the cells were treated with either hydrogen peroxide or any other means of oxidative stress. And these are the references I use throughout this presentation and also in my report. If anyone wants to go and check them out, I'll link them in the video description. Thank you everyone for listening.